Hello, everyone. Thank you for joining this Elveflow webinar today on flow and medium recirculation in microfluidic cell culture. I'm Lisa Muiznieks, and I'm a biofluidics application specialist at Elveflow. My colleague, Barbara Corelli, is a sales engineer for biology applications, and she will join us at the end of the presentation for the Q&A session. Before starting the presentation, a brief introduction about Elveflow. Elveflow is a startup company whose goal is to develop state-of-the-art instruments for researchers. We are researchers ourselves, and we aim to make microfluidics more accessible to specialist and non-specialist end users so that researchers from all disciplines that benefit from flow control can advance the state-of-the-art in their fields. In this presentation, we will describe how to move from a static cell culture platform, such as traditional culture flasks, to dynamic cell culture using microfluidics. We first introduce the advantages of adding flow to cells in culture and describe how to control the flow in a microfluidic chip. We'll then describe why you might want to recirculate medium and how to set this up. Then we'll finish with some recent applications of recirculation before our Q&A session. So firstly, why microfluidic cell culture? A major driver of using microfluidics for cell culture is to increase the physiological relevance of the cell microenvironment compared to the static and hard plastic surfaces of tissue culture flasks. Cell culture in microfluidic chips decreases the scale of the cell microenvironment towards the tissue scale. For example, the dimensions of a blood vessel. Microfluidic chips can have multiple channels that increase the dimensional complexity that can be achieved for cell culture, including growing cells on either side of a porous membrane or in a 3D hydrogel that mimics a tissue matrix, leading to the creation of more complex co-cultures with multiple cell types and of tissue interfaces. Improved in vitro cell models with greater physiological relevance aim to reduce animal models of drug testing. And of course, the addition of flow adds dynamics and mechanical stimuli to cells in culture. This is a huge benefit because fluid flow is a major physiological component of the body. The main sources of fluid flow are from the circulatory system that is comprised of blood that flows through vessels, from large arteries through an extensive network of small capillaries that reach close to every cell in the body, and back to the heart again through veins, and from interstitial fluid that surrounds cells and flows through porous 3D tissue networks. Excess tissue fluid is then absorbed as lymph into lymphatic vessels and returned back to the bloodstream. Microfluidics is perfectly suited to adding flow to cells in culture and can be tuned to match physiological flow rates, pressures, volumes, and length scales. The most straightforward benefits of adding flow to cells in culture are to enable a constant exchange of cell nutrients, efficient and dynamic gas exchange, as well as removal of cell waste products. On a practical level, this enables the researcher to automate medium changes and to culture cells in an experiment for days or longer. Other advantages of mimicking physiological fluid flow over cells in culture are to incorporate non-adherent, naturally circulating cells to an experiment, such as immune cells, red blood cells, or circulating tumor cells, for studies of cell rolling and adhesion, immune response, or tumor cell metastasis and extravasation. Adding a drug under flow conditions more closely mimics the residence time of the drug as it circulates the body through the bloodstream. Flow allows the researcher to incorporate more relevant length scales to test the effect of secretions from one cell type on a more distant cell type in a more realistic way. And flow enables control of molecular gradients for studies of cell migration, invasion, chemotaxis. Flow is also a key mechanical stimulus on a cellular level. Forces are abundant on cells and tissues in the body, but are largely absent from traditional static cell culture platforms. One example is shear stress, which is a force applied tangential to the surface on which it acts. For example, blood flowing through a vessel applies shear stress on the cells lining the vessel walls. Laminar shear stress affects cell shape, alignment, and distribution of focal adhesions, also gene expression and signaling cascades, and is a highly relevant addition for many mechanobiology studies. But how is the movement of fluid controlled? Well, in the body, the heart is responsible for pumping blood. In the lab, flow is most commonly controlled using syringe or peristaltic pumps, or else using a gravitational system like rocking platform. However, for increased stability and better response time, you can use a pressure-driven flow controller, like the OB1 from Elveflow. Pressure-driven flow works by controlling the precise amount of air pressure on the surface of the liquid in a reservoir. With increasing pressure, the liquid is pushed through the outlet tube. The greater the pressure, the faster the flow. The pressure controller is hooked up to liquids in reservoirs and connected to the microfluidic chip with tubing. A flow sensor can be added with a feedback loop, so you can control the flow of fluid by flow rate, not just by pressure. The ESI software allows you to choose the flow profile of choice through the chip, be it steady, pulsatile, or even custom, such as a heartbeat. But what if you would like to recirculate medium through your cell culture on chip, like recirculation of blood in the body, instead of having the liquid go to waste after it leaves the chip? There are many reasons you might choose to recirculate medium. These include practical considerations, like conserving medium if you run a days-long experiment or if you use a fast flow rate, or to reduce the amount of expensive reagent you might add to your medium. There are also experimental considerations, like being able to add non-adherent circulating cells to your experiment, or to mimic physiological multi-organ effects. For example, looping together cell models of different tissues, like a gut chip and a liver chip, so you can test the effect of a metabolite of one cell type on the behavior of another. Recirculation is also useful to more closely mimic the physiological residence time of a recirculating drug, and it also enables you to build up conditioned medium. That is, to enrich for molecules secreted by cells for off-chip analyses, or to complement the medium in a following experiment. So how do you set up a recirculation loop using a pressure-driven flow system? Firstly, you have to collect the liquid that comes from the chip into a second reservoir. You can't use this one. It's being pressurized to drive the flow. So the medium will recirculate between two reservoirs. As reservoir 1 empties, reservoir 2 is filled. But if you were to simply flow the liquid back from reservoir 2 in this configuration, the liquid would be pushed back through the chip in the opposite direction to which it came. To avoid this, we add a MUX-recirculation with a rotary valve. The valve has two positions for guiding the path of flow. In position A, liquid is pushed from reservoir 1 through the chip in the direction of the arrow and collects in reservoir 2. In position B, liquid is pushed from reservoir 2 through the chip in the same direction as before and collects in reservoir 1. The keys here are that recirculating flow is continuous, it's unidirectional through the chip, and at a known and controlled flow rate. Flow is also pulseless, and liquid is never constricted by a pneumatic action, unlike with a peristaltic pump. So now we bring you into the lab to demonstrate the connection of a MUX recirculation. Our workspace has the OB1 sitting on the table, and on top is a MUX recirculation. And we have a heated water bath next to this to hold our liquid reservoirs at 37 degrees. So the first step is to connect the pneumatic tubing for the pressurized air from the OB1 to reservoir 1. Then we connect the PTFE tubing for liquid from reservoir 1 to the MUX recirculation. Then we connect the MUX recirculation to a bubble trap. Now, a bubble trap removes air bubbles from the liquid as the liquid flows through it. Then we connect the bubble trap to the flow sensor. And the flow sensor is used to monitor and control the flow rate. And then the flow sensor to the chip. Here we are using a union connector in place of the chip for now, so we don't push air in the chip while we purge all the tubing on its first filling. Then we connect the chip back to the MUX recirculation. Then we add a loop piece between two consecutive MUX-recirculation ports. And then we complete the circuit by connecting the MUX-recirculation back to reservoir 2. And then we add the OB1 pressure controller to reservoir 2. And you can see with this setup, we've cut our tubing long enough to move a chip between a microscope and a CO2 incubator without having to disconnect the chip. So here is an example where recirculation was used to keep HeLa cells cultured in a microfluidic chip for over 50 hours. Live dead staining shows the majority of cells are alive, stained green with calcium. Finally, I'll spend the last few minutes describing some recent studies that have really advanced the state of the art using dynamic cell culture, in part due to the addition of recirculation. Firstly, coupling organ models for improved drug testing, then adding immune cells to study an immune response, and lastly, adding shear stress to study mechanosensors. So the first example used recirculation to link multiple organs on chip and test drug responses. This is a study from the labs of Anna Herland, Donald Ingber, and colleagues. And here, the authors used sets of organ chips, for example, a gut, a liver, and a kidney chip, or also a bone marrow, a liver, and kidney chip. And each organ chip is complex in its own right. It's comprised of two stacked channels separated by a semi-permeable membrane and populated with organ-specific cells on either side of the membrane. The organ chips were connected to a common medium, mimicking blood, that was recirculated through the lower or vascular channel of each of the organ chips in the direction following the red arrows. A drug was applied to the gut, mimicking oral delivery, or to the bloodstream compartment, mimicking an IV injection. The study showed that cellular drug responses for each organ compartment were similar to those seen in patients using oral nicotine or intravenous cisplatin in proof of concept tests. In particular, this configuration, including recirculation, provided more relevant residence times for the drug and clinically relevant drug concentrations. It enabled the testing of multi-organ effects, specifically drug absorption, distribution, metabolism, and toxicity. And it included scaling of organ size with blood volume ratios, all towards building a model of physiologically relevant organ responses for more accurate pharmacokinetic and pharmacodynamic drug studies. The second example used recirculation to add circulating immune cells to study an immune response in blood vessels. And this was a study from the lab of Carolyn Cheng in Utrecht. Here, endothelial cells and parasites were cultured together in a gel scaffold to create a model blood vessel with a perfusible lumen. Here we see HUVEC cells lining the vessel lumen in green. After 7 days, immune cells, in this case monocytes, were recirculated through the vessel in the absence or the presence of a cytokine stimulus, TNF-alpha. The attachment of monocytes, here in pink, to the cells of the endothelium was significantly increased in the presence of the pro-inflammatory cytokine after 24 hours. So this model captured an important first immune response at the site of inflammation. And this configuration provided the key ability to recirculate non-adherent cells for 24 hours. The third example used recirculation to study the effect of shear stress on endothelial cells that line blood vessel walls. This is a study was the work of Julia Mack and Luisa Iruela Arisp and colleagues. Here, the authors cultured endothelial cells in a microfluidic chip under high aortic stress to study the mechanosensor protein called Notch1. Notch1 is a molecule that is able to sense and respond to changes in the mechanical environment of the cell. Endothelial cells under high shear stress are known to elongate and orient in the direction of flow, as seen here. The cell-cell barriers are stained in green, and the cells are elongated and oriented in this flow direction. The researchers then showed that Notch1 was activated in endothelial cells in response to laminar flow proportional to the shear stress that was applied and was polarized to the downstream position of the cell. Shown here is Notch1 in red at the tips of the cells especially under conditions of the higher shear stress compared to the lower used here. So key findings were that Notch1 signaling was required for maintaining some of the main hallmarks of endothelial cell responses to flow and shear stress, including their elongated cell shape and alignment, suppression of overproliferation, and their anti-inflammatory profile that imparts an atheroprotective effect. That is, Notch1 responds to hemodynamic shear forces and protects the endothelium of the descending aorta from building up atherosclerotic plaques. With respect to the experimental setup, recirculation provided the ability to use high flow rates to test high shear stresses in their vessel model. So in summary, these studies show some exciting advances in the molecular understanding of healthy tissue function and disease states, and improved platforms for drug screening, in part through the increased physiological relevance provided by their dynamic cell culture models that incorporate flow and recirculation. So our key takeaways are that microfluidics is ideal to add dynamic flow to cells in culture. Flow can be tuned to match physiological flow rates, pressures, volumes, and length scales. And recirculation, for example, using a MUX-recirculation, adds versatility to your flow control and the ability to incorporate a range of physiologically relevant parameters that are not easily accessible by other methods. If you're interested in more details of cell culture, please check out our application notes online at elveflow.com. We have lots of information available on seeding, perfusing, and recirculating medium over cells in culture, as well as detailed protocols and lots of hints and tips. Or feel free to send us an email at contact@elveflow.com with any questions. We're always happy to talk and discuss your microfluidic setup. We also invite you to check out our online microfluidic calculator. It's a great resource to guide you through flow rates, pressures, and resistance to use in your microfluidic system, and to give you an idea of the shear stress inside your chip. So thank you. And now together with Barbara Corelli, we will look forward to taking your questions. Remember, please use the chat box to ask your questions, and we'll try to get through as many as possible. Thank you.
So let's see the first questions. Okay, so from Shojaish. Thank you, Lisa, for very attractive presentation. As it's going to be used for cell culture, I was wondering how do you sterile the tubing and fittings for cell culture? Yeah, I can answer as well. So our suggestion here is to replace. First of all, for every experiment that you do, the tubing is considered really a consumable, so you can replace the tubing. What you can do for also tubing and fittings eventually is to soak them in anuzyme, is a disinfectant, for 30 minutes, and then wash with water, ethanol, and air dry. So you can use this procedure. Otherwise, it's always recommended to change the tubing between each experiment. Next question. Thank you for the presentation from Becher. I'm using the max injection valve to set up a recirculation. I've been facing problems when seeding my cells with low flow, that the medium get clogged in the first connection inside the valve, so the flow does neither reach the flow sensor nor the chip. Have you seen that before, and how can I solve it?
Yeah, so you can, so I'm not clear if, well, yeah, you're seeding your chip first, I imagine, and then you seed with the concentration of cells that you want, let it attach in static conditions, if you like, and change the media by hand until the cells reach a confluence that you desire. Then hook it up to your MUX-recirculation. And actually, if you have a lot of dead cells that haven't attached, you can consider flowing a chip full of medium over your cells before you attach to the MUX-recirculation, or consider even adding valve to be a waste collection?
Yeah, in any case, actually, you can contact us and send us an email at customer@elveflow.com, and we will be glad to help you and give you some more technical advices. So I invite you to do that. Thank you very much for the presentation. Very nice. Thanks a lot. Any more questions? So thanks, a very interesting presentation. Can you actually control the shear stress in the chip not to damage the cells? Do you have any advice to compute the shear stress in the chip?
Yes, so you can estimate the shear stress inside your chip based on your flow rates using our microfluidic calculator. It's online on our websites. The link is also in this presentation, which I believe will be made available for you all to check again. Gives you an idea of what shear stress would be applied to the cells in the chip.
Yeah, so yeah, guys, feel free to use our calculator for playing with the pressures, flow rates, shear stress. It will be really useful to design your microfluidic setup.
Yeah, and of course, if you use less flow rate and less pressure, you'll have less shear stress. You can change the dimensions of your chip as well to play around with different shear stress in your cells, on your cells.
Yeah, that's true. Any more questions? So, a question from Zore. Thank you for the presentation. Can we inject air containing particles using MUX-recirculating system? Is it possible to trace particles motion within the chip?
Certainly. This is like adding recirculating cells, if you like. So here the key is you don't want the particles to clump together. So you want them to be dispersed. To trace the particle motion within the chip, well, I imagine that would depend on your chip. If you had the chip on a stage of a microscope and you could take a video, or if the particles were fluorescent or something, I imagine you could for sure think up a system to check that out.
Yeah, thanks, Lisa. So next question from Abbas, is there any difference between the microfluidic calculator and the experiment, or it is generally low errors? Well, of course, is an estimation that we give. So of course it is an estimation, I would say. So,
it's there as a guide to give you an idea, a fairly close idea, but it won't be 100% precise.
Yeah, it really depends. So, Shoresh, I've tried this suggested method for sterilization before, but is in the manual you have suggested that don't flow the water after ethanol to the flow sensor. It may result in precipitation of the salts inside the flow sensor, that it's in the circuit and it needs to be cleaned too. Yeah.
Yes, so this is only at the very end of your experiment if you are putting your flow sensor away, if you're finished with it. Then we recommend strongly after you've cleaned it to use either Helminex if you want to do a deep clean, or the aneurysm followed by water, followed by ethanol, followed by air drying if you're going to put it away. But if you are going to keep it in line in your system, water is fine. Let's see if I get the question right.
Yeah, don't flow the water after ethanol. Actually, yeah, the point is not just washing with water, if I understand correctly, but this can create some drops that can eventually leave some residues in the inner capillary of the sensor. So it's always better to flush with ethanol after and air dry, if we understand correctly. Another question from Agar, similar to the problem mentioned above, sometimes with two-channel chips, one flow circuit stops and both media flow in the other circuit. Any recommendations, knowing that there is no leakage in the chip itself?
It sounds like there might be. If one flow circuit stops and both media flow in the other circuit, then it sounds like in a two-channel chip, if I understand correctly, this is like a stacked channel chip with a membrane in between, there's probably some movement of liquid across the semi-permeable membrane. And maybe that's precipitated by one channel being blocked. And so the path of least resistance is actually to then push across the membrane into the other channel. That's, to me, it sounds like.
Yeah.
For recommendations, well, you can contact us for more specific details.
We can maybe look into it more closer if you send us an email at contact@elveflow.com. From Abbas, for the temperature, does it affect any sensors? I have seen in the video that we were putting the chip in an incubator. So as a general information that we give here, we put in the incubator only the chip, okay? So we just leave the tiny tubing that reaches the chip, let's say passing through the incubator, and so you can close it with the lid, because the OB1 and the flow sensors and the valve, since they have electronic inside, they have to stay outside the incubator. So that is the answer. Thanks a lot, Lisa. You're always really helpful and supportive. Thanks, Agar.
Thank you.
So from Andy, thank you for the great presentation. Actually, I'm planning to design microfluidic system, which includes movement systems such as stretchment. In such movement system, I think it might lead to unstable pressure due to the movement. Could you explain about this, please? Well, actually, we have experience with setup using a chip where we have two chambers, so that are with flow, and another channel of our controller that controls the stretch at the level of the membrane, just with air. And actually, we did not notice any particular problem at the level of the two fluidic channels of the chip. So you can definitely do that with some specific chip, of course, having two fluids flows and one channel of our pressure controller controlling there and applying the stretch at the level of the chip. Abbas, thanks a lot. Great presentation. Thank you very much. Any more questions? In any case, thank you, Lisa and Barbara. It will be great to leave the contact information here for further communications. Yeah, of course.
Sure. So this is Elveflow, contact at elveflow.com. Okay, thank you for moderator.
Thanks a lot. Agar, what is the difference between using MUX and not using MUX for recirculation, both using Elveflow systems?
Okay, so I believe you're using the valve system. So
exactly.
This is a question.
Yeah. Well, actually, both systems can work. Of course, it is a matter also of organizing your space in your bench as well. So the MUX gives a more compact solution, and I would say easier probably to set up.
However, the one difference between your system and using the MUX-recirculation is that if you have two channels in your chip that you want to recirculate, well, one MUX-recirculation is good for one channel, so you would need a second MUX-recirculation. So I think
you're right. It's true. That is the difference. Soren, thanks a lot. A great presentation and Q&A session. Zore, is it possible to explain about tracing tiny particles motion in the airflow within a microchannel? How it is possible? I'm not sure.
Do you mean in the air, or do you mean in the liquid phase?
Maybe you can explain a bit, or just
in the air. I'm not sure it's possible.
I'm not sure either. You can in the air. I'm not sure. Maybe we can look into it.
Maybe we can look into it if you write us an email, please.
We can discuss this further, I think. I'm not sure. So
Okay.
if agar, yeah, is there are flowing cells in the tubing, will this clog the system?
Not necessarily, but perhaps, yes. If they are dead cells that are very sticky, or if they're clumped, then they have a good chance, yeah, things that are aggregated have a good chance to clog the system. Depends a little bit on what is the most resistive part of your system, so how thin, the thinnest inner diameter that you're using. But they may.
It might happen, yeah. That they
Especially
could aggregate.
the dead cells.
Yeah.
Yeah, if they're live cells and they're added intentionally. You have a comment about blood?
Yeah, well, actually, I mean, with, for instance, with the circulating blood, the idea is to use some steering or like making, and adding eventual anticoagulants, for instance, for blood, like heparin and so on, to keep for instance, the blood fluid. So yeah, to avoid, of course, these clogging issues. So Joseph, Xavier, is it possible to mimic the nebulization using Elvis' system lung on a chip platform?
Yeah, so I think you're referring to adding vacuum to one of the channels to stretch a membrane. Yes, for sure.
Or probably create a sort of air-liquid interface, probably. Is it what you are meaning? Maybe like flowing air of a specific concentration or a specific mixing. So that is possible, knowing that actually the OB1 needs an external pressure source. So usually you use air or maybe nitrogen, CO2, but you can make your own gas mixing that you want to apply. This is the one that will pressurize the fluid in the reservoir and that will reach also the side of your chip where you want to put this gas in contact with your cells, for instance. Yeah. Agar, great ideas. Thanks. Joseph, thank you. Well, I think for today, if there are no more questions, we are done. So guys, thank you very much for participating to this webinar. And if you have any questions or if you want to plan a microfluidic setup for cell biology, in your biological lab, you're welcome to write us at contact@elveflow.com, and we will be happy to help you and design the system with you.
Okay, thanks a lot.
Thank you very much. Have a good day.